My thesis is titled The Litter-Associated Benthic Macroinvertebrate Communities Along the Elwha River. So essentially, I studied the tiny organisms living at the bottom of the Elwha River and discovered what they can tell us about one of the most ambitious ecological restoration projects in US history, which was the removal of the Elwha and the Glines Canyon dams. Um, I want to start out with a quote by E.O. Wilson. Invertebrates are the little things that run the world. Most people overlook these organisms, and yet they perform the foundational ecological work that um, sustains river food webs. When we ask what dam removal does to a river, we need to evaluate how it's affecting the invertebrates. So the Elwha drains from the heart of the Olympic Mountains and flows through the lower Elwha Kalam Reservation down to the Strait of Juan de Fuca, as you can see here on this map. Just below the Glines Canyon Dam right here, 80 is the Olympic National Park boundary. So about 83% of the Elwha River watershed is within the national park, making it very distinct from other rivers in the United States. In 1913, sorry, it's a little different I'm not presenting. Oh man. Okay, well, this image is supposed to go away and it's not going to in this view, sorry. In 1913, the Elwha Dam was constructed, and in 1927, the Glines Canyon Dam was built 8.5 miles upstream from the Elwha Dam. Both dams were then removed in 2011 and 2014, respectively. And at that time, this was the largest dam removal in the world. About 20 million cubic meters of sediment, a century's worth of sediment flushed downstream in a geologic instant. 20 million cubic meters, by my calculations, is about 10 lumen fields. That's the Seahawks Stadium full of sediment that came down the river. All right. Okay. So why monitor invertebrates? Freshwater invertebrates are doing the unglamorous but essential work of breaking down leaves, cycling nutrients, and feeding just about everything above them in the food web. Remove invertebrates and the whole system starts to unravel. Many benthic macroinvertebrates are also incredibly sensitive. They suffer, the ecosystem follows. And here's the kicker. 5% of US dam removals have generated published science. So we're essentially removing the dams faster than we're studying the effects of those projects. So my research question is, how do litter-associated benthic macroinvertebrate communities vary across the restored Elwha River 2 to 4 years following dam removal? Benthic macroinvertebrates, I predicted that the upstream sites would be healthier, more diverse, and more sensitive than the sites closer to and below the former dams. So in late August 2016, Carrie Leroy, Sarah Morley and company deployed leaf litter bags at 20 sites across 5 regions. The litter bags are mesh packs with dried leaves. For this study, we used dried maple leaves with equal weights and 5 replicates for each site. The image that you see here with the leaf litter bag is not ours. I borrowed that from Oregon State. But ours had full mesh all the way around to allow water to flow through. The sites began just inside the Olympic National Park, as you can see on the map. Um, and they continued along the main stream, up the tributaries, and down all the way to the newly formed estuary at the river's mouth. All bags were harvested on September 18th, 2016, and there was one small hiccup. Locations 9 and 10, which you can see here on the Aldwell Reservoir area, were accidentally combined, so our sample size was reduced to 19 instead of 20. Before we move on, I want to make something very clear. So the tributaries, all of my figures coming coming up are color coded and they move from upstream to downstream. So tributaries are the streams that feed the main stem of the river, and they're going to be a light blue. Then you have the dark blue is the what we're calling the middle Elwha, and it's on the main stem. Mustard is the Elwha Reservoir region. That's the sites there. And then we have olive is the lower Elwha, and turquoise is the delta. Okay, so this is what I did. The sampling team pulled invertebrates from all five replicates litter bags per site. Invertebrates were preserved in 70% ethanol following the collection. Approximately 20,000 individual benthic macroinvertebrates were sorted morphologically from leaf litter and detritus using dissecting scopes. So this image in the just above counting and sorting is what it would look like as a raw sample. So you can see leaf litter in there. You see little pine needles, some decaying detritus. And in there are still insects. You can see like there's a little mayfly here and maybe a little fly here or a midge. And we had to carefully pull each one of those out of the detritus without damaging them. It took a very long time. Each organism and every bag was sorted, counted, and identified. So you can see here, this is us sorting them by morphology into these pools of the sampling trays. Um, Now, they were all identified, 81% was identified to the genus level. And oftentimes, identification began by observing diagnostic characteristics, such as caudal fins, as you see here on the bottom left. They're like tails. The shape and placement of external gills. On this one, you see that white fuzz is actually external gills coming out between its legs. Abdomen, leg, and feet features. So all the little features of the legs and the feet, as well as physical traits along the ventral side of the thorax. So you see all these little nooks, these little divots inside of here. All of those have names that help us identify the insects. In short, insect taxonomy took a lot of patience and incredible, like fine, detailed scrutiny. It often felt like I was an insect dentist because I had to learn every nook and cranny of every mandible and maxilla, as you see in this top right, all of these mouth parts of the insects. Endless teeny tiny animals under a microscope, hundreds of hours, and my favorite time in the lab ever. After all of that, I input all of this data into spreadsheets, and this is what we found. So to begin, I want you to recall, please, the upstream to downstream color coding of our graph. So you have the tributary, middle Elwha, Aldwell lower, and delta, all color coded upstream to downstream. This is where the previous Glines Canyon was located on the map, and this is where the Elwha Dam, again, removed in 2011 and removed in 2014. So this is Shannon's diversity figure. Shannon's diversity tells you how many types of organisms are present and how evenly distributed they are. So the higher the value, the healthier the river. So the tributary sites had a mean of 1.7, whereas the lower Elwha scored the lowest at 0.62. These are the communities here that absorb the most disturbance, the lower Elwha and the Elwha Reservoir, and they had the lowest Shannon's value in the data set. So years after the dams came out, again, these were sampled in 2016, are still organizing around whoever could survive. Percent EPT, EPT, Ephemeroptera, Plecoptera, and Trichoptera, mayflies, stoneflies, and caddisflies. These are the organisms that are most sensitive freshwater invertebrates. A high percent EPT is a healthy river. So the tributary samples had 63.5% EPT. That's a pretty healthy tributaries. And then, as expected, in the delta is brackish, where insects don't usually live. And the delta had 0.1%, probably an outlier. However, the lower Elwha, which is still freshwater, but at this time was blanketed in sediment, had 2.2% EPT in the samples. Community tolerance values. This is this figure. CTV is essentially how tolerant a community is to pollution or disturbance on average. So a low score has a sensitive organisms are dominating. Here you can see in the tributaries again, you got those sensitive communities. And the high score is a more tolerant organisms are dominating, as you see here in the delta. Tributary sites scored a 4.27 CTV and the delta scored a 6 CTV. So this gradient almost mirrors perfectly the percent EPT result. It's a different metric, but we're seeing the same story. These chironomids, midges, they have a CTV, a tolerance value of 6. And then the radix snails also have a tolerance value of 6. And this is what a community looks like after a significant disturbance, very tolerant. And these are organisms that we found from the lower sites. So I do want to mention one caveat. The 66% of the individuals at one of the sites in the Delta were excluded from the CTV because we couldn't identify them finely enough. These were mostly worms, which are among the most tolerant taxa in freshwater. So the true delta CTV is almost certainly much higher than we're reporting here. Okay, so this figure shows results of a multivariate analysis called NMDS. You can think of it as a map of how similar or different the invertebrate communities are to each other across the 5 regions of the river that we sampled from. So the points that cluster close together have communities that look alike, as you see here in Aldwell Reservoir. And the points that are far apart have communities that look really different, like you see in the tributary. The tributary sites are undisturbed and they're forested streams and they sit in one corner. The middle regions lay pretty distinct from each other. And then the delta is out here on its own in the opposite corner. So the river is writing its own story across the ordination space. And that story follows the river downstream. So it's intact at the top, disturbed at the bottom, and then the river is still finding its way in the middle. And what strikes me the most about this figure is that the pattern is emerging across every metric I measured. Shannon's diversity, percent EPT, evenness, NMDS, they're all showing the same gradient. It's true, it's consistent, and it follows the river. So that's one of the key findings. The gradient was consistent across every metric. That convergence is probably the most important analytical result of my study. Another surprise that we saw was the Aldwell Reservoir was consistent with early successional recolonization. What that means is that many of the invertebrates were arriving opportunistically with the community composition dominated only by a few tolerant species. There were some caveats in the study. There was no pre-removal baseline specifically for litter-associated invertebrates. There was only a single harvest date, which is essentially just a snapshot in time. And these results only describe the litter-associated invertebrates, not the full benthic macroinvertebrate assemblage. This is a term I coined in my thesis because it kept coming up. The term is legacy disturbance debt. The ecological consequences of long-term impoundment that a system is forced to comprehensively reckon with at the moment of dam removal. So, what I'm saying here, the idea is that a century of damming doesn't just pause a river, it reorganizes it. When the dam comes out, the river didn't start recovering from zero. It started from wherever 100 years of damage left it at. In a pre-dam removal study by Morley et al. On the Elwha River, the midges dominated the benthic macro community in the lower Elwha before either dam was removed. So it was already showing high tolerance values. The 2016 communities that I sampled weren't solely a product of dam removal. They absorbed a sediment pulse on top of an impairment that was already decades deep. So yes, 20 million cubic meters of sediment buried the substrate and thus excluded the sensitive taxa. That's crucial. But the chronic piece started in 1912 when the dams were established. The removal of the Elwha's dams was an act of ecological restoration on a scale rarely attempted. But as Rachel Carson observed, life adjusts over millennia. It is not designed for the pace at which humans impose change. The benthic macroinvertebrate communities documented in my thesis are not a simple story of failure or success. They are a record of where the river stood one moment in a recovery that will likely take decades to unfold. The most important thing we can do now is keep watching. So none of this happens without a lot of people and a lot of love for tiny animals. I'd like to express my deep gratitude for my mentor and my advisor, Carrie, for her endless patience and willingness to work with me. And many thanks to my sorting team, Sarah Morley's team, the Leroy Lab, and of course the Lower Elwha Kalam tribe for fighting to restore their home river, and to all the invertebrates, fish, and other organisms whose lives were shaped or lost during the time of the dams and their studies. This one's for them too. Any questions?